We see them everywhere. Exosomes are now widely advertised, from skincare to regenerative therapies. Yet even in medical communities, they remain mysterious, misunderstood, and often misrepresented. That's why we've turned to one of the world's leading experts to help us separate fact from fiction. Meet Dr. Aijun Wang, Chancellor's Fellow and Professor of Surgery and Biomedical Engineering at UC Davis, where he also serves as Vice Chair for Translational Research, Innovation, and Entrepreneurship. He is surely one of the most prolific innovators in the field of extracellular vesicles and nanomedicine. With over 180 peer-reviewed publications and over 20 patents and a track record of translating therapies from the lab to the clinic, Dr. Wang is on the front lines of unlocking how exosomes can be used, not just marketed, as powerful tools for healing. In this episode, we dive deep into what exosomes and extracellular vesicles really are, how they're made, what they do, and where the science is going next. This isn't hype. This is the real story from a pioneer building the future of regenerative medicine one vesicle at a time. Please welcome Dr. Aijun Wang.
In the not-too-distant future, a revolution is brewing, one that will change the very fabric of our existence. It's a tale of cells that hold the key to unlocking the mysteries of life itself. Let us venture into the unknown, for in this age of stem cells, anything is possible.
I've seen some beautiful...
You are a sensation around here. People are, after your talk, everyone's talking about your talk.
You know, I recently attended a conference another conference because I like to collaborate with different people. I was given a new title, Mr. Collaboration. I'm seeing lots of people interested in collaborating with engineers to make new products for patients.
We talk about stem cells on this podcast regularly, and there's so much information out there that's so confusing. And a lot of times people confuse things like PRP, exosomes, and they dump all this into the world of stem cells. And so I am often faced with the challenge of trying to explain, well, what's the difference? I mean, they're two completely different things. But I thought, you know what? I'm going to give this hard job to somebody who can better explain this than me. And I'm going to pick your brain, Doc, and you're going to explain to me you know, like we make exosomes, and so let's take a step back from exosomes because there's a whole process behind. First starting with like how you make them, because you need cells.
Yep.
Okay, so then you take cells and you feed them, and when you feed them, just like humans, you know, we eat.
Yep.
And we poop.
Yep.
Is that a fair explanation? Okay, so the cell eats food and then it poops out stuff.
Yeah.
And then you take this collective food and poop combination, and that is essentially what we call, in the industry, we call conditioned media.
Yep.
Okay. So conditioned media has now evolved into, because you need this concoction to then further purify.
Yep.
Is that the right word? Purify?
Yeah. Purify or isolate. Yeah.
Isolate.
Okay. Yeah.
Okay.
I think you're getting absolutely right. Yeah. Cells, all the cells are consuming materials. They're going to, have output as well, just like everything else.
So why is that output valuable?
Yeah. So what we call, exosomes or other particles secreted by cells, we call them extracellular vesicles as a general, broader term.
Is that different from exosomes?
It is actually different. I think that's one of the things we should clarify.
Okay, let's do that.
Yeah. So extracellular vesicles are a broader term. So all the vesicles secreted by cells are considered extracellular vesicles.
So that would be the highest sort of category.
Yep. Like a much broader term than exosomes. So exosomes, from biogenesis of exosome pathway. So it's a very specific type of extracellular vesicles. But there are many other type of vesicles secreted by cells. We all class, because you can't technically isolate exosomes from cell culture media only. but you can't avoid other vesicles because they're all in the same media and they're all similar. And the traditional, well, we should really think about exosomes and extracellular vesicles they're related, closely related, but they're different. Depends on how you isolate them. When you say purify or isolation of those EVs, extracellular vesicles from culture media, we usually take either size-based approaches or specific capturing mechanisms to get a subpopulation of those kind of vesicles. So, but the size-based, either by ultracentrifugation or other, ultrafiltration, size exclusion chromatography, or other methods to separate those vesicles from the rest of the conditioned media. Because, there are lots of things in the conditioned media. You only isolate those vesicles from the media, and the rest will be separated.
So, so simply put, you have this concoction, and there are various methods to pick a very specific population of compounds that you're looking for.
Yep.
And I guess it depends on the disease that you're studying that you would want to pick that compound.
Yeah.
But how many compounds are we talking about? So when you say there are these from the top extracellular vesicles, how many are we talking?
So extracellular vesicles are those vesicles, right? Usually nano-sized particles, usually from 50 nanometer to a couple hundred nanometer.
So it's defined by size.
Yeah.
Oh, okay.
Yeah.
Okay.
I mean, that's one way to define that.
Okay.
There are other ways to depends on the sort of the mechanism of action. We talk a lot about that. How when you use something as a treatment, you want to make sure it's designed for that indication, right? So you have a specific scientifically designed to that treatment. So for example, it could be like a protein A based isolation. You can pull down a protein A binding particles by using that antibody, for example, to pull down protein A based vesicle, but nothing else will be pulled down. So that'll be one way. That'll be very specific subpopulation of EVs. But I think the general concept of isolating extracellular vesicles usually refers to size-based separation.
Okay.
So you get a range of nanoparticles from culture media and deplete all the other free proteins and metabolize other molecules.
Then exosomes fall under the extracellular vesicles. So is there a certain size?
No. So exosomes actually they're smaller. So some people call them small EVs, small extracellular vesicles. Because lots of other vesicles are much bigger than exosomes. Like apoptotic bodies, for example, when cells are in apoptosis, they can secrete lots of bigger vesicles. They are actually they're quite different from EVs by biogenesis pathway related features and also composition. So this is important to think about one is the biogenesis of EVs, extracellular vesicles, and also the other one is the composition. So I talked a lot about the heterogeneity of these extracellular vesicles, which is very important because from biology, this is just natural, right? All these vesicles are in the media, and cells are pretty smart. They respond to the environment. Right. If you culture them in different environment, they're gonna do different things. So they're gonna secrete different vesicles if you stimulate them. So that's just biology. That's what it is.
So right? these vesicles, and can you give me an order of magnitude, like how many we've talked about MSCs. Take an MSC as an example. How many extracellular vesicles would you suspect they secrete? How many different kinds? How about that?
How many different kinds of extracellular vesicles? Well, there are a few. So kinds is not that many.
It's not that many.
Yeah.
What's in the billions then? Oh, that's the quantity.
Numbers, yes.
That's the numbers.
Yeah.
Okay.
Because cells they keep secreting things.
Right.
If you feed them enough, they're gonna secrete stuff, right? Of course, you need to. There's a digestion, consumption as well. But eventually, if you isolate the conditioned media, add fresh media, and then you keep adding and collecting. So you're going to get lots of vesicles from the culture media.
So proteins and cytokines,
Yeah.
where do they fall into the EVs?
Okay, very good question. Usually proteins, cytokines, they are free proteins. They're free proteins. If you run regular ELISA, right, you only detect using antibody, you only detect those free proteins usually.
Okay.
But there are proteins that are associated with vesicles.
They're like, would it be accurate to say that the EV is like a car?
Yeah, like we talk about cargo, right?
Oh, okay.
Lots of things inside the vesicle. Can
Okay.
be lots of proteins, lots of lipids. I'm just from a composition. Lots
Okay.
of proteins, lipids, nucleic acids, and metabolites. Usually, those are the bigger categories of compositions inside the EVs. So some proteins can be associated with vesicles either on the outside. We call them like a protein corona, or other ways proteins are associated with the lipid bilayer of the vesicle, or inside. I think the proteins could be they could be inside as well. So lots of and they are very different. So unlike synthetic material, you can control those lipid nanoparticles, for example, relatively speaking, or polymer-based particles. They are more homogeneous in a way.
Okay.
You can make them homogeneous at least relatively easily. But EVs are so like bulk EVs can be just take them all together. You can say, okay, composition A is a percentage or whatever unit you use to quantify, but you can't really say for sure which one it's hard because they're so small, right?
Right.
Each individual one is relatively hard technically to characterize.
Right.
So, but we're trying to characterize them more and more carefully with the more cutting-edge technology, single nanoparticle characterization tools. But still, it's not that straightforward, not that easy.
So, if we haven't lost you yet, the beauty of these compounds is that you have these cells that are essentially factories that are making these compounds, right? And so that's the novelty of it. Is that so? What did we do before the cells? Can you make these compounds in a lab? Like synthetically, can you make any of these compounds in a lab?
Compounds in the lab,
Or extracellular vesicles? Can you make any of these things
well,
in
you're
a lab?
asking the right person.
Okay, yes,
we are making
that's
those.
what I'm asking.
Yes, yes. It's not easy because cells it takes lots of steps and compositions in culture media to make a natural EV.
Yeah.
Right? It's pretty complex. Proteins, lipids, metabolites, lots of things are in there. So that's why it's not easy to make.
Okay.
But we are taking some synthetic biology-based approaches to make EV-like construct, right? To mimic the native natural derived EVs and to mimic like just part of it. You can't make still, at least at this moment, it's not that easy to make completely synthetic, exact natural derived EVs.
So I'm guessing that the reason why you're going backwards is because you found something that the cells are secreting that is helping.
Yep.
And you're saying, okay, now we know what this is.
Yep.
So we have to find a way to mass produce it to make a medicine.
There you go.
Right?
Yes.
So
yes.
we're going to
and
make it
be
in the
more
lab.
homogeneous, right?
Exactly.
Yeah. So you can make, exactly, you start from biology. Okay, this is, because EVs are mediating very important cell-to-cell interactions. Because one cell secrete EV, there's a reason why they're doing that, right?
Right.
The cells are always communicating with other cells by either like contact-mediated interaction or like remote interaction by secreting proteins or other molecules being uptaken by the other cell. So that's communication. But EVs is just like that. So EVs are secreted by one cell and then it will be uptaken by the other cell. That's a way for cell-to-cell communication. So, but there are lots of things. When they do this, it's not like only secreting one thing from cell A to cell B. It's always associated with other things. So when we say, okay, EVs are good for this and for that, so we possibly only say a fraction of the story, right? That's the one protein is associated with that vesicle, and that seems to be functioning that way. But there are lots of other things associated with that. It could be good or bad, right? It could be like synergistically supporting the same function, direction function, or maybe not. Maybe like, so it's just like a complex. We're trying to dissect this. Right. So maybe one protein is mediating one function or a set of functions. I mean, as you know, one protein can mediate lots of functions sometimes.
So does the, when you're identifying these EVs, you said there's a handful of EVs. Like how many?
Oh, how many different kinds? So not that many. So I don't think it's there are lots of new types of extracellular vesicles are being identified.
Oh, really? So it's an ongoing thing right now.
Yeah.
Okay.
So exosome is being one of the vesicles, EVs, that has been very much discussed or studied and investigated because that's the majority of the extracellular vesicles that are biologically functional to mediate cell-to-cell communication. Because there are other vesicles too.
So is it then not, the importance is not so much the protein that it's carrying, or is that the important part?
So usually we believe proteins are very important
okay.
for EVs. That's why we use lots of ways to characterize the protein quantification or presence of a specific protein on EVs as a biomarker. Sometimes we do lots of proteomics to study EVs because there are many different kinds of proteins. Some proteins are higher in expression, and we're trying to correlate that to certain functions. And some proteins are known to be functional in certain, function. We believe even those proteins are associated with vesicles, they're going to be maintaining the same function. So, for example, in our lab, we study lots of neuroprotective function of extracellular vesicles. So stem cells can secrete those vesicles to help neurons to survive. We found actually from just cell culture and animal studies, you can identify those actually is pretty consistent if you have the stem cell-derived extracellular vesicles cultured in a specific way, isolated in a specific way. And those neurons in apoptosis with those stem cell-derived extracellular vesicles, and they can help neurons to survive from apoptosis. So we have associated those phenomena with specific proteins that we know they're related to like a neuronal survival.
So let's say you get to a point where, I imagine this world to be as like as mysterious as the genome, how genes work together. I assume extracellular vesicles also work together.
Oh, yeah.
And so to know the combination of, maybe it's not one EV, maybe it's a combination of 4 EVs that will produce a certain effect. But all of that, I think, will, with AI and different tools, I think we're going to learn a lot of things faster.
Yeah.
So if we were going to produce and by we I mean the industry, if at some point they discover something, or whether it's a single EV or a combination, and then they are successful in creating a synthetic compound that actually works, how do you see that being delivered? Is this something you can take in a pill? Is this something that you have to get?
That's also a very good question. I think it will be easier compared to cells.
Right,
because these
all
are
the
nanoparticles.
logistical.
Yeah. So like IV injection, because the cells are because of the size, and if they're bigger cells, just physically, because after IV injection, many of them will go to certain organs. It's hard to change. But nanoparticles, actually, they have a better circulation capability, I would say. They can go to different places more freely because they're not so much limited in terms of their size. Yeah.
So that's really interesting. So for cells, we depend, at least when we deliver intravenously, we are always depending on the innate homing capacity of the cells, that their ability to go and navigate to areas. I heard in your talk that you can engineer these EVs to do the same.
Yep.
To go to a specific location. So, but if you don't do that, do they just go everywhere?
They also have some preference, apparently.
Okay.
Because I think just like other particles, they go to circulation because they'll have lots of molecules on the surface, that just naturally derived vesicles because of the parent cells will make their own EVs in their own way, right? So cell A derived EVs may be very different from cell B derived EVs just because they are from different like parent cells. So their destination by distribution profile could be very different. Yeah, because the presence of specific proteins on the surface. What we're trying to do is to engineer the surface so you can guide them to a more specific location, organ tropism, cell type specific targeting. Those kind of things can be achievable actually if you have a known target for a specific disease, right? You want most of them to go to a certain type of cells, certain location. Upon injury, no injury, you try to target basically. You could treat those EVs as some other like a physical particles. Essentially, you can add different molecules to the outside, just exogenously, right? Different from endogenous expression. You can add those molecules to the outside, like chemically, for example, physically, for example, and that will guide those nanoparticle distribution. You can change it.
So if I were a pharmacist, and we have in modern medicine all of these different compounds that we know of today, the EV world, would you say like in the next 10 years, next 20 years will further expand that option, that pool of options?
I think it's a very exciting time because biology, we know so much more about those extracellular vesicles now because of lots of the new cutting edge technologies. We can characterize them better. We can track them. You can see where they're going, right? So that's very important. It's not a black box anymore. Previously, because of the limitation in technologies and the size of the particle is so hard to study, make them very hard to study. So, but nowadays, actually, with all these new technologies, you can actually characterize them. You can study them with quantitatively with lots of new approaches. I think it's a time to really think about like EV biology and EV medicine in a more rigorous, scientifically rigorous way. You can quantify them. I think you can it's exciting. I think more things will come for using EV-based therapeutics.
And you see the future as doing it that way, where once we use the raw materials that the cells have provided as sort of a base, we're using that sort of as base knowledge. And then working our way backwards will be sort of the way the industry will go or the field will go. You think?
There are many ways the industry will go. I think there are so many ways. Different type of cells you can precondition or treat the cells differently. You get different kinds of EVs. And again, I think we always talk about mechanism of action. Right? You have a disease, what's needed for the treatment, for example, to be effective. And then you can design your product based on that mechanism of action. And then you can treat. I think you're right. EVs are made by cells. The parent cells are kind of functioning like a factory, right? They keep pumping out. If you treat the cells nicely, they're going to do the nice thing to you. They're going to produce all those extracellular vesicles. And then you can isolate them. There are lots of ways to isolate them with good quality control. And you can characterize them, and then you can really I think there are lots of other things that need to be done. Storage, transportation, just like other drugs or formulations.
But it's certainly a lot simpler than the cell world, because the cell world, cells have such a short time frame of once you isolate. Once you isolate the EVs, you could even freeze dry them and
Technically.
Do you think that stability is compromised when you freeze dry?
The lots of research in the field is going on right now. Apparently, it's important. Compared with cells, live cells are so. They're sensitive, right? They can be killed. They can be changed. They can, because they have DNA. They can they can replicate. There are lots of safety concerns as well if cells survive and they do. Tumorigenicity, for example, possibly all the cells can be transformed. But unlike cells, EVs are relatively, they're nanoparticles, right? They don't have their own replicating capability. You don't have as much, but they have lots of things inside that. So not to say they're always safer than cells, but technically they're more amenable to off-the-shelf storage or application between patients maybe. So
because they have no DNA.
Yeah, they don't replicate. Right?
They
Unlike
can't
cells,
be rejected.
they proliferate. They cannot be rejected. Is that
Can
what
they
you
be
said?
rejected?
Well, there are lots of proteins. They could be rejected.
Oh, they could.
Yeah, there are lots of proteins,
okay.
right? Proteins may induce like reaction to the immune. Depends if they can be recognized by the immune system. If the immune system is recognizing that as a foreign material, and what the immune system will do. So it's not like they're always being immune protected. There's no. I mean, some cell-derived EVs are more immune protected than others.
I see.
Yeah, but there are lots of proteins. Imagine if those proteins are triggering immune responses that can cause immune rejection.
Okay.
Yeah.
So, on TV and on the internet, when you read about things, people selling exosomes or doctors offering exosomes, generally, is that a mixed product? In general, I know everyone has different stuff, but in general, is it a mixed?
So it depends. I think it's getting. I mean, the science is advancing rapidly with new technologies and new characterization tools. I think now we, at least from the EV biology perspective, we know much more and better about what they are, what they look like, what they're going to do, just based on characterization, right? I think having better characterization of those vesicles, extracellular vesicles, is critical. I don't know, there are lots of different ways to say what you have, but you have to have the data to support what you have. I think
so you should be, if you are, ideally in the future, if you're getting an exosome product, you should know what's in it. Is that?
Oh yeah.
That's okay. Oh yes.
Very
okay.
very important.
All right. Okay.
Oh yeah. I think from the regulatory approval perspective, you do need to know what's in there, how much do you have,
how much of it is in there,
and how consistent it is between batches.
And that
right?
and that's the definition of a drug. I mean, you have a known dose, known compound. It's not a mystery. And so,
absolutely.
If you are seeking exosome treatment and you see a product that doesn't have, you don't know what's in it, and they're just telling you there are billions of things in there, that should be red flags.
Absolutely. You should ask for certification of quality.
Yeah, and what's in there.
Those kind of what's in there, what's known, how much safety data do you have. All these are important information to have. I think if it's from the regulatory approval perspective, all these information will be needed for approval.
So are exosomes currently regulated?
Yeah, it's a biologic.
They are. It is a biologic.
It should be a biologic.
If it has a claim, right? How are doctors selling it now? I see it everywhere that exosome treatment is available. I guess they're without a claim, maybe.
Okay.
I don't know. I've seen it.
I don't know very much about the industry side.
Okay.
It depends on the application, I guess. If it's a medical use, I think you're gonna have to have a
BLA.
Yeah.
Yes.
Yeah.
If you produce, let's say, conditioned media and get exosomes, you would suspect that every bottle you make, even if you're starting with the same cells, is different.
Yeah. Well, I think collectively you should have a quality control.
Like a standard.
Standard. You're going to have a standard.
Yeah.
And release criteria, right?
Sure.
As a product, you have to have a set of criteria you have to go with.
Mm-hmm.
And so, so you can't say they're always the same, or cells are always making the same exosomes or EVs. That would not be the truth. I think you have to quantify the cell, and culture condition need to be very well controlled, and product need to be very well controlled.
Yeah.
So they can be very different.
Sometimes I find myself staring at the bottle of conditioned media, just staring at it, wondering, I wonder what's in there.
What's in there?
What's in there?
Well, there are lots of microscopic approaches
Things.
nowadays. You can just zoom in and in and see what's in there.
Does it always amaze you that there seems to be no end? That
I know. Yeah, it's pretty amazing actually. Now we know so much more, but we still feel we don't know enough.
and these super tiny things are regulating our bodies in such a fine-tuned way. Very powerful.
Lots of very powerful information there, and proteins. One protein can trigger lots of downstream pathways, right? Lots of microRNAs in there, so that way we know for sure. And some of them are master regulators of certain pathways. It can be very powerful. Yeah.
So if you had your crystal ball and you were to predict the future, at what point in time do you think exosomes will be a part of our medical future?
I think very soon.
Oh, really?
There are lots of ongoing clinical trials, very well-designed studies. So I'm pretty excited about that, actually.
Okay. Well, we'll be watching your work and following that. Thank you so much.
We'll be happy to share whatever we discover in the lab.
Well, good luck with all your work. Thank you.
Thank you.
Human